Put your cell phones on the silent mode. The Institute of Technology and Immunobiologicals, Rio Manguinhos from the Oswald Cruz Foundation, has the pleasure of welcoming you at the 8th International Symposium on Immunobiologicals 2024. The goal of the event is to integrate scientists, some researchers, and institutions towards the development and production of immunobiologicals, in addition to providing visibility and providing capacity building to professionals and promoting also opportunities and partnerships in the biotechnology sector, stimulating an environment of scientific development and technological growth at the national and international level. We would like to thank the following sponsors and supporters: Sanofi, Promega, GSK, Biotech, Sativa, Sykes, Cepheid, Beckman Coulter, Bionovus, AstraZeneca, Getúlio Vargas Foundation, and Croda. We would like to recommend that you gain access to the application of the event directly through our website or through the area of participants that is being shown on the screen. Through this application, you're going to be able to ask questions directly to the speaker and to participate in the set of customer surveys, including a professional profile, so that all participants at the event will get to know each other, exchanging information and having access to all the works that have been enrolled. And I would like to recall that our event is taking place in hybrid fashion, and you can follow the whole program live through our application. You can also participate, asking your questions here in the audience or online through the chat room, and we'll make sure it will get to the chair of the panel. The public here in the audience have simultaneous translation equipment, and for the online audience, all you have to do is click on the language you prefer on the icon in the lower portion of the video. Our lectures will be recorded in our application. You can listen to them at your whenever you feel like it. We've had a campaign, our friends in the south of Brazil, and 1 kilo of non-perishable foodstuffs or feed for animals, hygiene items, cleaning items. You can bring them. If you haven't brought these items, there is a little market. There's a sugarloaf market right next to us. I think it's very important for us to undertake this joint action, and you can leave your donation at the tent we have right next door. Now, starting the presentations, the posters, I'd like to welcome on stage Marisa Oliveira Ribeiro. She will present the topic Respiratory Virus Panel and Respiratory Virus Panel 2, Bio-Manguinhos Molecular Typing Assay for discriminatory diagnosis of respiratory infection. Please. Good morning. Do you all hear me? Well, let's go. My name is Marisa. Good morning. I'm a biologist. I work at the molecular diagnosis laboratory in Manguinhos. We develop kits. I'm going to present the respiratory virus panel 1 and 2. It's a molecular typology tool that is a discriminatory diagnosis tool for respiratory diseases, the main respiratory viruses, which have a clinical importance for Brazil, and immunological importance for Brazil, and are the target of molecular diagnostics. On this panel are SARS-CoV-2, S2, which is the which caused the latest coronavirus pandemic, influenza A and B viruses that lead to seasonal respiratory illnesses, the metavirus, human metavirus, which leads to common flus, the respiratory virus that is present and circulating among our population, which may lead to severe bronchiolitis in young children and may lead to death as well. The rhinovirus, which is commonly circulating in Brazil, and it's very characteristic of hospitalized kids. It's very important because it affects children at the hospital, and that may lead to more severe diseases than the adenovirus, which is present throughout the year. It's not seasonal. It causes common cold and flus, but it's present throughout the year in all seasons. As for clinical aspects, I can tell you that all these viruses co-circulate in the population and they present similar symptoms. Just from the clinical diagnostic, we can discriminate what's the cause, the virus that causes the respiratory disease, and people can deal with the infections easily without the need for specialized clinical assistance. But these viruses can lead at times to severe illnesses that may lead to death. And that's very worrisome among children, pregnant women, the elderly, and health professionals. As for prevention, what the WHO recommends is personal hygiene, avoiding contact with people with symptoms to avoid contamination, and prevention through vaccines against SARS-CoV and seasonal flus. As for diagnostics, Bio-Manguinhos, which is the arm of Fiocruz that develops molecular kits and vaccines, has quick tests, but it has acted also in the development and standardization and distribution of molecular tests for respiratory viruses that has intensified because of the pandemic. We had to develop, molecular tests to detect mainly SARS-CoV. And this latest test was developed on the left as a molecular test that discriminatory test for influenza A and B and SARS-CoV-2. This test is being distributed to breastfeeding women. A lot of the patients that had respiratory symptoms and had undetectable results for MB, AB, and SARS-CoV-2, we noticed that they have symptoms that may be caused by another virus. And at this point, we saw the need to develop a molecular panel, discriminatory molecular panel that would be more embracing, encompassing, and that could take care of other viruses that could lead to the aggravation of other illnesses, FB, AB, and SARS-CoV-2. We've had a long-term partnership with a benchmark laboratory at IOC led by Dr. Marila Siqueira and Dr. Fernanda Mata, and we have listed the viruses that would be most prevalent among the population. We've listed the viruses that would make up a new panel for respiratory diseases. We have two multiplex reactions. One of the VR1 reaction detects influenza A and B, pneumovirus, human pneumovirus, and then adenovirus, respiratory illness, rhinovirus. And these two reactions that take place at the same time have the internal control, the room RSP. These are respiratory reactions, discrete and sensitive. They're part of a multi-centric study. We have distributed these reactions, over 100,000 reactions, as a whole sum of research. It has not been registered at Anvisa. These reactions have been developed by the laboratories around Brazil. We have had the chance to develop a cooperative study with a laboratory at the Medical School of São Preto under the leadership of Dr. Mauricio Nogueira. This group has a great deal of expertise in the development and production of vaccines against arboviruses. In this cooperative study, we had access to 48 symptomatic patients that had been previously tested in-house through the CDC protocol at the medical school for SARS-CoV-2. This SARS-CoV-2, these patients, 10 of them had positive results for SARS-CoV-2. When they were tested with the in-house method, the same 48 patients were then tested for VR1, VR2, which is the Bio-Manguinhos test. And what happened was we had 100% agreement, and the results of positive results of 10 positive samples from on the in-house test were positive from VR1, VR2. Of the 10 positive samples, there were 38 samples that were not detected for SARS-CoV-2 through the medical school test. Of these 32 samples, 11 were detected for other respiratory viruses when they were tested for a VR1, VR2 panels. Now with this, through this investigation with VR1, VR2, they were positive for other viruses. They were distributed. Of these 11 other samples, 2 were positive for influenza A. One of them was positive for metapneumovirus, human pneumovirus, and then RSV, 3 for rhinovirus, and 3 for adenovirus. And to our surprise, another sample was positive, triple infected. Among these positive samples, one of them was positive for SARS-CoV-2, COVID-2, and RSV at the same time, concomitantly. This is important because we know from the literature that co-infected patients may have the aggravation of the disease. It was important to know what when it was amplified, we detected also for other respiratory viruses. The sample, it was a 9-month-old child with moderate symptoms of a respiratory syndrome. Here we can notice the amplification curves of the 3 viruses and their respective CTs through the PCR, real-time PCR test. Those respiratory curves that leave no doubt as for the real diagnostic, and that was important for us to be able to visualize and know that these results were discriminatory and definitive because the 3 viruses had been clearly identified. We did a sample later. The week we were present at the medical school, we got the epidemiological report from the region, and we ascertained that at the time we were doing the cooperative test at the medical school, the most prevalent viruses in that town were SARS-CoV-2 and also rhinovirus. And we can notice also that the age bracket that was most affected was children under 12 years of age, under 12 years of age. We can identify a triple infection in a child under 12 years of age from the viruses most prevalent in the town. These are the data from April. They tested 450 samples from the in-house test, and they were not detected for SARS-CoV-2. These undetected samples were tested on our syndrome panel, and they had only been tested for SARS-CoV, and they were positive for 335 samples, and the other respiratory viruses that are part of our syndrome panel were detected here. And see in the graph, in the bar graph, among these samples, the most prevalent one was RSV. As for the conclusions, we can see that the panel was extremely capable of detecting and discriminating respiratory viruses that cause respiratory infections that are most prevalent in Brazil. We have observed also that it is able to discriminate the virus that's causing specifically that syndrome, and it may discriminate also what we know that some are more severe than others depending on the age bracket and the patient conditions. That's very important to our clinical studies. It's important from a laboratory perspective. It's important because it's 2 reactions that detect 7 viruses all at once. You don't have to do it in-house individually. You can detect it through the syndrome panel. And we also have the expectation that this panel will be certified at Anvisa by the first semester of this year. We'll be submitting the dossier of certification by the middle of the year. And once it has been certified, it will be a very important clinical tool and to detect epidemiological infectious diseases. I'd like to thank AMAL, the Medical School of São José do Rio Preto, which was very helpful in our tests, and the smallpox and mumps laboratory of respiratory viruses of the IAC. Thank you.